Okay. So we're going to now do is shift gears and return to the digestive system, which we introduced during the lab. And again, at this point, we have 2 weeks left. We have 3 main systems to cover. We have to finish the digestive system, the endocrine system, and the reproductive system. And we'll see the endocrine system and the reproductive system have a lot of overlap. And when we get into the details of the reproductive system, a lot of it is going to be following the different hormones and their cycling up and down and how they are working with your gamete production and how the gamete production and the gonads are creating hormones that are then going back to your pituitary and all of that. So although our first introduction to the hormones, I guess our first introduction to hormones was epinephrine when we did the sympathetic nervous system. But we're going to see that control of the digestive processes relies very heavily on hormones as well. The first place people first discovered hormones was actually trying to understand the coordination of digestive system activities. Like, how does your gallbladder and your pancreas know that there's food in your small intestine? You know, it turns out that it's hormones released by the small intestine that go through your bloodstream that then end up binding to receptors in your pancreas and your gallbladder. So, I don't know if it's ironically, but interestingly, the hormones are critical for the functioning of the digestive system. All right. Digestive system. All right, so again, we've introduced this when we started the lab last time. The basic idea here is we have food. Food is stuff that is usually used to be the body of some other living thing, right? It used to be a cow or a fish, or maybe it's some kind of plant. It used to be a tomato plant, or it used to be a wheat plant, or it used to be I don't know, some kind of seaweed. It was something that was some other living organism. And that living organism was made up of the same basic molecules that we are, carbohydrates and proteins and fats, but they're assembled in the patterns that were appropriate to be a cow or a apple tree or whatever. And so what we have to do is we take that in and we have to break it apart into the basic building block molecules, which we then use and pattern into our own structure or break down for energy. So the first step is always going to be kind of break down of, and I'll say food, which is all that stuff you're putting in your mouth that has nutrition, again, used to be some other living thing, into building block molecules. Right, we're going to take proteins and turn them into amino acids. We're going to take carbohydrates and turn them into monosaccharides. We're going to take fats and turn them into fatty acids and stuff, just like we saw in the lab, or you saw in your pre-lab. We'll see in the actual data of the lab today. The next step is you actually have to bring it into your body. All of this breakdown is occurring officially outside of your body. When we think about the digestive system, it kind of looks like this. Here's like a person. And if we think about the digestive tract, it starts here at the mouth. There's the esophagus. There's the stomach. There's the small intestine. There's the large intestine, rectum, and finally the anus. This whole thing here that I just drew, this is called the intestinal, also known as GI tract, also known as the alimentary canal. And basically the food that you're eating and putting in your mouth here, it is going down this system. It's in your stomach, in your intestines. This is where it's breaking down. This is where all the breakdown's happening. Whatever is left is going to ultimately come out and be your poop here, the feces. But the food and the breakdowns is never actually in your body. It's always in this tube that's running through you. People talk about, topologically, we are basically a donut with a hole at the top and the bottom and a tube connecting the two holes. So the second part of this digestive system is going to be bringing the nutrients across the walls of this GI tract into the body. So that's the absorption of the nutrients. Some of it's going to be really simple. You've seen already, like transepithelial transport to bring glucose into your body and absorb into your blood. We saw that in the first few weeks of class, right? We saw crossing from the lumen of the intestine across the epithelium and get them to go into a capillary. You know how that works with the carrier and the secondary active transport with the symports and blah, blah, blah. Looking at the absorption of fats will be a little trickier because fats are not going to dissolve. They don't go through carriers. So it's going to be a little more complicated looking at absorbing fats from the intestine and getting them in your body. But kind of back to our kind of big picture here. One, breakdown of food into building block molecules, and two, absorption. So we'll be looking at both of these. We'll look at the processes to break your food down and how and where those occur. And then we'll also be looking at absorption, how you bring the nutrients into your body as well as where that occurs. Most of this is mostly in the small intestine, particularly the jejunum, which is the kind of middle part of it. And there's a little bit of absorption of like water and some drugs in your stomach that we'll talk about. There's absorption of water and maybe some vitamins down in your colon. But for the most part, when we talk about the lion's share of absorbing nutrients from the GI tract into your body, it's going to be mainly in the small intestine. Okay. So, our lab today is all about breakdown. Let's start this just talking about some of the basic thoughts about breakdown. If anybody has connections to the people who do the user interface for Zoom, if you could send me their contact information. I don't like some of their newest revisions here. It is definitely non-intuitive to make it do what I want to do. So mechanical versus chemical breakdown. Mechanical is just physically ripping things apart and crushing them and basically increasing surface area for the ultimate chemical breakdown. This is going to be use enzymatic breakdown. Mechanical breakdown is taking in fact, I should just do it. Hold on. All right. Right. Here is some yummy cracker, and it's got lots of nutrients, but they're all locked and baked into this little disc. We know there are enzymes in my saliva, but they're going to have trouble really doing their job if all of this thing is still baked into this big hard wafer. So the first step is mechanical, like chewing. All right. So am I actually breaking apart like starch into glucose and maltose right now just by chewing?
No.
No, I'm just like crushing it, increasing the surface area. Now, as it's all smushed in, now I got my saliva really permeating it. Now that's kind of what's starting to happen.
The enzymes are breaking down the starch.
Exactly. Now I'm starting to get that that chemical breakdown in my mouth, as well as the amylase from my saliva is actually starting to hack apart the bonds between the glucoses and the starch. So chewing is mechanical breakdown. When you swallow stuff and put it in your stomach, you've probably noticed if you like eat a meal and then you barf pretty quickly afterwards, you can recognize exactly what you ate. You just see chewed up food there. You don't actually see goo. You'll actually see, oh, look, there's my chewed up spaghetti or whatever. Right. So once the food's in your stomach, there's more mechanical breakdown. We're going to talk about the walls of the stomach has this extra layer of muscle even. It's just going to be smush up, smush down, smush up, turning your food into this paste. That's like more mechanical breakdown, this kind of kneading as your stomach is just smushing up and down and up and down. So mechanical breakdown is important. By the time you get deeper down into the digestive system, then everything is all just kind of a goo. And at that point, it's just kind of purely about the chemical breakdown, the enzymes dismantling the different molecules. While we're at saliva, we start here. What else is saliva helping me besides starting to break down the starches into smaller chains?
Making it more moist?
Yeah, exactly. Imagine if I just chew this up, I just try to like swallow dust. I would not work, right? So part of our lab is actually going to be looking at your saliva has two components. It has what we call the serous fluid, which is actually the amylase, has the enzymes, but there are also mucus cells in your salivary glands making mucus. So part of your saliva kind of show. Right? It's called a bolus. Bolus means a little ball. It basically turns what used to be dust into a nice slippery little ball that's going to be easy to swallow without choking. So saliva is going to be important for the beginning of our breakdown of starches, but it also has that mucus in there, which is going to bind your food into the little bolus so you can swallow it. Oh my God, I've made a mess. All right. All right. So I've kind of already introduced the idea of the alimentary canal. Or GI tract. So we're going to spend a bit of time looking at that in more detail. And then the other thing that is part of the digestive system are all the accessory structures. Basically all the different glands. We're going to see the liver and the pancreas and stuff like that. The salivary glands feeding into there. Your teeth. The gallbladder, which is not quite a gland, it's more like a little sac that stores bile. It's also etc. So two main things, a GI tract, which is the tube. We have to understand the tube since its properties are going to define a lot of the functioning of the digestive system. And then we'll bring in, as appropriate, various glands in your teeth and all that kind of stuff. But I would like to just introduce the GI tract as a more generic thing right now. Like on Thursday, we'll be going through it and looking at the specializations as you go from region to region. Obviously, the oral cavity is different than the throat, is different than the esophagus, is different than the stomach, is different than the small intestines, different than the large intestines, different than the rectum and the anal canal. So they all have specializations for the different things that are going on in the different regions. But there are some characteristics that are the same wherever you look. So that's what we're going to look at right now. The kind of more basic layout of the GI tract. It's basically a tube. The lining of the tube is a mucosa. It's just a wet epithelial-based membrane. So that is going to be, maybe I'll make it, oops. It's mainly going to be simple columnar epithelium, except if you're like in the mouth or the esophagus where it's like stratified squamous to deal with friction. This is that epithelium that we're going to have to pull the nutrients across, right, in order to absorb our food. This is that simple columnar epithelium that I drew in our really early discussions of transepithelial transport, right? And the inside of this is the lumen. The lumen just means the internal part of this. That's where all the actual like breakdown is happening and all the food stuff is mixing with the enzymes and smushing around, and ultimately once things break down all the way to basic building blocks, that's when you can, bring them across the wall and you'll have the capillaries and blood vessels sitting on the other side to actually absorb the nutrients. All this stuff we're going to be doing in our lab in the test tubes. It is kind of modeling what would be happening within the lumen, either in your mouth, whether you're chewing stuff with saliva, or in your stomach where things are smushing inside the middle of your stomach with the acids and the pepsin, or in your small intestine where there's a mixture of the digesting food and the pancreas and the lipases from the pancreas and the bile coming in from the gallbladder. But all the stuff we're doing in the test tubes is modeling what would be happening in the lumen, and again, after the breakdown, then you'd have to pull stuff across into the blood vessels on the other side of the epithelium. That's the absorption. There are a variety of subtleties to the different layers and of the mucosa and all that. You don't need to know that for this class. The only other main layers you need to know the Muscularis Externa. Muscularis Externa is smooth muscle that lets this tube either squish or kind of expand. So let's draw that. Smooth muscle cells that make a round so these are supposed to be smooth muscle cells. So this is going to be the circular layer. This is all what we call the Muscularis Externa. Circular layer, if these cells squeeze because they're going around circumferentially like this, they're going to squeeze the lumen. They'll squish the tube shut. So it'd be like if you put your hand around the tube and squeezed it. The other layer are called the longitudinal layer. They're going kind of the long ways along the tube. They're kind of more outside. The circular layer is kind of more inside, and then on the outside of that are the fibers going the long way. These guys are going to basically pooch the tube out. So the circular layer will squish the tube, the longitudinal one will kind of open it up. So using this smooth muscle, you can either squish the tube closed or you can kind of pooch the tube open. So these are going to be how we're going to be manipulating the food that's in the tube. Should also mention, except for like the esophagus and certain areas that are retroperitoneal, most of this is all coated with serous membrane that's called the peritoneum. The peritoneum is just maybe I should just write it out using my typing tool. So the peritoneum is just the serous membrane. Everything is in this double layer basically with fluid, so everything slips and slides. So even though your food is constantly getting smushed and these intestines are moving and squishing and smooshing, they're not causing friction between each other because they're all coated in this serous membrane. In anatomy, you get way more into detail about the peritoneum. The peritoneum has special areas where it's double layered called mesenteries that are kind of supporting the intestines. There's the greater and lesser omentum, the transverse mesocolon, and this and that. You don't need to know that for this class. For this class, you should know that there's a serous membrane that envelops the majority of your digestive organs, and it's called the peritoneum. While we're talking about the Muscularis Externa, I said there are some fibers that basically squeeze the circle to squish the tube. Some fibers squeeze the long way to kind of pooch out the tube. There are two kind of basic patterns of contraction, and then there's lots of things that kind of grade back and forth between them. But you should know the two fundamental patterns of contraction of this muscularis externa that do two different things. One is if you want to move things along the tube, you use peristalsis. The other one, if you want to mix stuff up that's in the tube, you just need to kind of smush it around because it's digesting and you need to just agitate it, kind of like shaking the bottle or whatever. Then you have segmentation. So let me do those both explicitly. So this is going to be patterns of muscle contraction in GI tract. First one is going to be peristalsis, which I'm sure everybody has heard. This is for propulsion. If you're trying to move things along, the classic place you see this is like in the esophagus. The esophagus is basically pure peristalsis. So if you swallow some piece of food, basically as you swallow it, the circular muscle above it is going to contract. Below is going to relax and pooch out, which is going to result in this bolus, in which we talked that ball of food, ending up getting squished down a little further down. And then once it's a little further down, we squeeze on top of where it is now and we relax and pooch out underneath it. And now a few moments later, we're watching it and it's further down. So that's all peristalsis is. It's just that squeezing from the top, relaxing from the bottom, and keep it moving along. Again, for propulsion. Like this is why you can lie on a couch, horizontally and have someone feeding you grapes. And when you swallow the grapes still make it to your stomach. They're not getting there by gravity. They're getting smushed along by this peristalsis. One of the things we're not going to do in the lab that we do sometimes is you can actually listen. You can swallow and put a stethoscope on your entrance to your stomach and time how long does it take from when you swallow to when the food actually reaches your stomach, right? Because this person looks weird. Person swallows and gets down to their stomach. From the moment you swallow, that swallowing is called decluttition. That's a word you probably should know. Like the first part of swallowing is voluntary. You actually are shoving the food to the back of your throat and kind of stretching out the top of your pharynx. But once the swallowing gets started, then it becomes an autonomic reflex. It happens automatically where these muscles start constricting and continuing to push the food down to your stomach. It's about 5 to 7 seconds. From when you swallow the food to where it lands and plops in your stomach. It's possible sometimes that the food gets stuck and the wave continues on. You've probably all felt that of you swallow some food or some pill and it's like, it's still there. Something didn't work right. When that happens, the body will automatically initiate subsequent waves of peristalsis, which will then hopefully catch the thing and continue moving it down towards the stomach. So that's peristalsis. Peristalsis to move things along. In the esophagus, it's pure peristalsis. The only reason to have an esophagus is move things from the mouth down to the stomach. Later on, we still see peristalsis, because things ultimately have to make it all the way to your anus and make it out of the digestive tract. So there's going to be peristalsis all the way along to move things to get to the other end. But for the rest of it, it's not going to be pure peristalsis. It will be mixed with what we call segmentation, which is the mixing part of the muscular contraction. So for mixing, we have something called segmentation. So segmentation rather than peristalsis. And there, the best way to draw it, assume this is your intestine here. Let's use some yellow food globs and some blue food globs. And now, instead of squeezing on the top and relaxing on the bottom, we're going to be squeezing in order to just mush things around. Squeeze here and here and here and here, relax here and here and here and here. And so then when we look at the next moment, whoops. Oh my God. There we go. Now our blue thing's half here and half here and here, and the yellow thing is half over here and here. And and then for the next one, we're gonna do it again, except now we're gonna squish here and relax here. And before you know it, in fact, I'll use green. It's just smushed. It's the blue and the yellow all smushed together. Again, imagine you had a long sausage. Right, and it's just going smush, 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 back and forth, smush, 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 smush. Right, so that's segmentation. So you're basically putting in the digesting food, you're adding all the digestive enzymes and juices, and now what we got to do is basically just keep mixing it up and agitating it and letting the enzymes do their work. So segmentation versus peristalsis, which is moving things along. Okay. So the last thing that I want to add in about the GI tract is what I think is super cool. Here's the GI tract. Let's put in there's muscles, whatever. It turns out that there are these nerve networks embedded within the walls of the GI tract. There's a few main ones. There's one nerve network that's actually right under the mucosa called the submucosal plexus of Meissner. And then there is another one that's actually in between the muscle layers of the Muscularis Externa called the Enteric plexus of Auerbach. So these are nerve networks. And they create what's called the enteric nervous system. Right, so don't worry about the anatomy of it so much. What I care more about is that you understand that there is this system of nerve networks within the wall of the GI tract. And everything we've been talking about so far when we talk about reflexes, we always talk about, there's stimulus, there's integration, and there's some response. And where does the integration always happen so far when we've been talking about reflexes?
Spinal cord?
The spinal cord or in the brain, but the CNS for sure. So it turns out in digestive processes, there's a whole bunch of reflexes that never involve the CNS at all. It just is integrated within the enteric nervous system. Sometimes they call it like the primitive gut brain, right? Because your digestive tract is actually doing its own thinking, figuring out this is what's going on, this is what I need to do, and doing it. If you think about this enteric nervous system, what kind of stimuli, what kind of receptors do you think it uses to kind of monitor? What kind of effectors, what kind of things do you think it uses to actually act on things? Like if you were in control of monitoring and controlling digestive processes, what kind of receptors would you want to have?
Stretch receptors?
Absolutely. Right, that's how you know if your stomach's getting filled up, or if there's something stuck in your esophagus, or if there's a big glop in your intestine to squeeze to the other side during segmentations. So the stretch receptors, these mechanoreceptors that let you know how full or empty different parts of the tube are going to be critical. What other kind of receptors?
Chemo?
Yeah,
Chemical?
exactly, chemoreceptors. And chemoreceptors, very much like what we're going to see in our lab, right? If you want to work with the digestion of carbohydrates, first you need to know that the carbohydrate's there, so you know what is the appropriate enzyme to mix in. So chemoreceptors, for substrates of digestion. I want to know what kind of molecule is there, so I know what kind of enzyme I'm going to need. And then I also want to know, is the breakdown happening? Is it done? So I can see the substrate disappearing. I can have for the products of digestion. Just as we see maltose shows up, meaning that the carbohydrate is broken down, right? Your digestive tract has similar things to see what's there and is it broken down? Are we done? Can we go to the next step? So chemoreceptors, also for pH, right? We've been talking about how important pH is for the effective action of these enzymes. So obviously chemoreceptors that monitor the pH to make sure the pH is in the right zone for whatever enzyme we're thinking about at the moment. And then in terms of what kind of actions, responses, do you think we should be able to control with this enteric nervous system?
Glands?
Totally, glands, right? We're going to be able to release different digestive juices, enzymes. We're going to see bicarbonate juice to change pH of things. So like glands for like digestive juices. And we're going to see also endocrine glands as well for hormones, because the hormones are going to be important in the control of the digestive processes. So exocrine glands, glands that just make stuff and smoosh it out, but also endocrine glands that release hormones that have more kind of nuanced control of other things. So glands, and what is the other main effector we usually talk about? If I wanted to do peristalsis or segmentation, what would I have to be controlling?
Smooth muscle?
Yeah, totally. The smooth muscle in the walls to kind of smush things around. Right? So just kind of giving you this sense, you have this concept of short reflexes. Integrated totally in the enteric nervous system. So there are reflexes where the alimentary canal, the GI tract, is monitoring this stuff, is doing the calculation, what do I need to do, and having some response. And it never uses the brain. That is what we mean by a short reflex. It is possible that the brain is involved. There are also long reflexes that involve the CNS. Right? If you just think about food, you're going to actually start salivating and having food, like digestive juices spewing into your stomach. We're going to talk about that later. We talked about sympathetic, parasympathetic. Sympathetic will actually inhibit digestive processes. Parasympathetic will stimulate digestive processes. So the brain can be involved. And if the brain is involved, we call it a long reflex. But there are plenty of examples where the brain is not involved. So it's a trippy thing if you think about it. Serotonin is one of the main neurotransmitters involved in this enteric nervous system. So just being aware that your gut has its own little kind of primitive brain controlling a lot of the processes that are going to go along without having to bother your CNS. So that's what I want to kind of just lay out. We've kind of given you this, I think enough to go on. So when we kind of hit the ground on Thursday, we can then actually talk about what's happening in the mouth, in the esophagus, in the stomach, in the small intestine, in the colon, in the rectum and the anus, and talk about the absorption at the different levels, and then a few other things. But that's all lecture for now.